of you who are in the class for the industrial biochemistry. Okay, welcome once again to SBT 458, Principles of Industrial Biochemistry. This is our first class for the semester, and we'll do a brief overview of what we'll be doing in the principles of industrial biochemistry. Oh, okay. Mary says you are 20, and currently you are only 3 people here. Anyway, we are recording the proceedings, so later I will share the link with you so that you can share it with the class. It's good that I taught you biochemistry last semester, because if you look at the curriculum for the industrial biochemistry, you realize that there are some overlaps with what we did in biochemistry. That is understandable because, yes, it is biochemistry, all right, but this time around, it's applications in industry. So there are certain things that I would only refer you to go and revise them. But what I try to do is, to make it more interesting and also for you to get more out of the course. I'm still working on, developing it in such a way that we'll focus on some very specific applications, to make it more interesting, more practical. Even though it's a theory course, you should be able to relate it directly to what goes on in industry. And also because there are lots of developments going on. So along the line, when I chance on any new things, technologies and things like that, I will also, bring them forth to share with you. For now, let's go over the course outline and see the things which are, within the course that we'll be going over. Okay, more people are coming now. Okay. All right. So, what we'll do is, first of all, there is some aspect of microbiology. And so you have to also go back to the microbiology you did, HND, and now BTech. You bring all of these things together. So we will look at applications of microorganisms in industry. So that would be in different industries. It can be food industry, food and beverage, like breweries, bakery. It can be the pharmaceutical industry. So we'll look at these things, and even looking at manufacturing, leather, textiles, things like that. Biocatalysis, we did a lot on biocatalysis last semester. That was the part of general biochemistry we did on enzymes. It's repeating here, so I will not want us to go over some of those things. But what we'll do is we'll take a look at those things that we studied. We see how they are applied in industrial engineering, for instance. And then uh, you also will be looking at scaling up of um, processes like fermentation, which is actually some aspect of um, the metabolism we did. Remember, we did metabolism of carbohydrates, and when we were talking about uh, catabolism, the breakdown of say sugar, uh, say the process of glycolysis, Krebs cycle. We did mention that that process of the Krebs cycle is in aerobic condition. In an anaerobic condition, you find that there is some formation of ethanol, formation of lactic acid. We did look at some of those things. So how these things are applied in different industries, like for instance, in the production of say yogurt, for instance, or uh, say BS or alcohol, ethanol, or even in biofuels, in the production of bioethanol, for instance. So we'll look at things like that. Um, Renewable energy. We'll be looking at, say, energy production from biomass, and also uh, something pertaining to biophotolysis. And that's almost like talking of how microorganisms, uh, or let's say plants, for instance, with the process of photosynthesis, for instance, could be converted into some energy and stored. It could be the use of algae, not not the say big trees, plants, but like how sometimes just in the growth of say algae in some aquatic systems, could be used for the production of energy, for instance. Um, some production of maybe some food additives, some uh, biomolecules. It can even be production of some enzymes. Last year, for instance, what we did was we actually did some virtual excursions as well to look at to look at videos of how processes go on in specific industries. Apart from the practical course that you do for this, for the practical course, they went for some excursion at I think the waste management facility of University of Ghana. So they went there, and so they could relate some of those things. But I think that was the only place they went for the practicals. But in the theory class, we actually watch videos of different industries to see how some of these things that we are talking about are actually practically implemented. And then we will also look at say waste, the waste management, different aspects of this, looking at sewage, wastewater, and even some look at what we refer to as bio extraction of metals. So that's a form of say recycling, and at the same time sort of a degradation of maybe it can even be some toxic metals that you we implement some microorganisms to break down some of these substances into less toxic ones, things like that. So there is some even interconnection between here. There is some interconnection between the course with toxicology, slightly like that, and much more industry into the extraction of metals like uranium, for instance, how it is extracted using some of these microbiological techniques. Some metals that are found in waste, how these can also be broken down to make the waste less toxic. So generally, these are the things we'll be looking at. And then we met last semester, so you already know how we go about our assessments, because we're having these lectures online throughout. Well, this semester, hopefully, getting to the end of this month, maybe coming to Ghana, so maybe we can have some face-to-face lectures along the line before the semester ends. At least if I'll be around there for some time, so we can have some face-to-face lectures. But for now, as time goes on, I will get some self-assessment questions for you to help you to understand the material better as you read, as you watch the videos. You try to solve some of those questions, and then it is out of those questions, just as you saw the last time. Out of those questions, we had some of them coming to the exams. So I mean, it's the same. It's the same modus operandi, the way we go about it. All right. I could see that Mary is typing, so maybe she has a question. So let's see what she's typing, and then we can respond to it. All right. So that's about the course outline. It's not anything complex per se. So I mean, it's straightforward. And because we have been together, last semester, you already know how we go about our classes. As a class is ongoing, all your microphones, cameras are off, but the chat is available, so you can use the chat if you want to ask any questions. And then at the end, if you also want to speak, you can raise your hand. I will unmute your microphone. I will enable your microphone so that you can unmute yourself, and then I will just call your name, then you can speak, then I respond. Okay, Mary has written and was asking if is the class always going to be on Tuesdays? All right, so I think I had some chat with you over the period. I think I'm getting to understand that the evening class for that I think maybe the principles of industrial. I don't know whether I don't know whether it's the industrial biochemistry people. There is only one student, which I don't think that it is really. I don't know which word to use to be teaching only one person in a class in the evenings. So if it is so that there's only one person in the evening, then it will be better for that person to come and join you on Saturday. Is it Saturdays? On Saturdays, so that for this class we can have it on Saturdays because I think let me look at the timetable again. For SBT 458 Industrial Biochemistry, we can have it on Saturdays. So I mean, last Saturday I was actually getting ready to be with you, but the the Microsoft Teams was just hanging. I just couldn't enter I couldn't log in. I couldn't do anything. So that's why we couldn't meet last Saturday. I think after today, we can be meeting on Saturdays for the industrial biochemistry. But the uh, toxicology, that one too, I don't know if there are evening students because as at now, I have not heard anything from evening students for industrial biochemistry and for toxicology. All communications I've had so far is with only you, the weekend students. So I think when I was chatting with you, I was contemplating, or we were talking about the two classes for toxicology. SBT, for the toxicology one was supposed to be in the morning, and then the other was supposed to be in the evening for two groups. Like you have a group for industrial biochemistry, and then a group for, I think, environmental chemistry also. And one of those groups, I don't know whether it's the industrial biochemistry that's supposed to be in the morning, and then environmental chemistry in the evening. So if you look at that timetable, it was like I was going to be teaching the whole day from morning till evening on Saturday. And those two classes, the toxicology in the morning and toxicology in the evening, just wasn't really convenient in that sense. But you have not been able to um, solve that problem so that we could combine those two classes on Saturday. Instead, you made me understand that you will be joining the evening class, which means that for toxicology, you want to have it on Tuesdays. So fine, we can have toxicology on Tuesday and have industrial biochemistry on Saturday, if that's okay. Mary and the class. If any the rest of you also listening, if you have anything, you can write in the chat so that because we have to take that concrete decision to know when we are going to do these courses. For toxicology, it's clear to me. I want to get that confirmation. It's clear to me that for toxicology, we are going to be having it on Tuesdays. But for this class, this one we are having now, which is uh, industrial biochemistry, we will have it on Saturdays. Is that okay? By the way, is there anyone in the class right now who is an evening student? If you are an evening student, kindly confirm in the group for me to see. Let's see that we have some evening people here, or else it means that this is just a Saturday class that I'm meeting, and there is no evening student, which means the decision we take is for us uh, weekend students only. That's it. Okay, so Alexander says the weekday is not convenient due to work schedules and other family commitments. That's why we chose the weekend option. So that is it. I understand that. And so that is one of the reasons why I really would want us to have the courses on Saturday. But if you want to have the courses on Saturday, you should be able to combine the toxicology class. The toxicology class should be one class. It's not convenient to teach toxicology in the morning, like 9, to some few students, and then later again, another small number of students in the evening. Okay, Mary says she knows only one evening biochem student. And so that is what Emmanuel also told me the same thing. The rep for the I think environmental chemistry also wrote me a few minutes ago that he, to the best of his knowledge, there was only one student for the industrial biochemistry. So if you know that person, I think you should communicate with that person to get in touch, because it appears that because she's alone, she doesn't even know what to do. So that we can let her join the Saturday class, uh, the industrial biochemistry class on Saturday. So that one we just know that industrial biochemistry is on Saturdays, and that's it. Now let's come to Alexander's concern that the weekday is not convenient, which means the toxicology to be doing on weekdays is not convenient for him. So this brings us to the question again. Is it possible to combine the two toxicology classes, the morning and the evening? All right, what I'll do now is I'll just let me enable all the microphones so that, because this one is a discussion, so anyone who wants to say anything, the person can speak, and then we hear. All right, I've allowed all your microphones. So if you want to say anything, you can raise your hand. I will call your name, and then you can unmute yourself and speak. Mary, you are the class rep, so I don't know if the other class rep is also here. And I put a question on the floor. What is preventing us from combining the two classes? Is it is it that impossible that we cannot combine the class which will be in the morning? I mean, I'm talking about the toxicology, where you have one group, maybe the industrial biochemistry group in the morning, and then the environmental chemistry group in the evening. Is it that it's just impossible that we cannot combine the two classes in the morning because you are few? Your microphones are enabled, so I want to hear from you. Don't just keep quiet. If you keep quiet, we can't solve the problem. This is interesting. You are not being helpful. So, I mean, how can I help Alexander? Uh, you know, understanding his concerns. I understand it. So how can I help if those two classes cannot be combined? Because it's just impossible to teach toxicology in the morning to some five students and later another six people in the evening. Yes, as your micro is enabled, so you can unmute yourself. Your microphone is off, but you can unmute yourself. Just tap on it and unmute yourself and speak. Nobody is saying anything. Very interesting. All right, so Belinda says joining in the morning may not be convenient due to work commitments. We would prefer to participate in the evening instead, if possible. You are talking about a toxicology on Saturday. We are talking about Saturday, and you are saying work convenience, or you are not an evening student. You are not a sorry, you are not a weekend student. What I'm talking about is the original timetable where on Saturday, toxicology is on the timetable twice, one in the morning, one in the evening. And if you are a weekend student, uh, why are you talking about work? Because you are supposed to be in class weekends. All right. So since nobody is saying anything, I think I'll just we just do a brief overview of the industrial biochemistry, and then uh, maybe you can have some few minutes break, and then I will continue with toxicology. That is, those who are doing toxicology can use the second link for the toxicology. It's very interesting, your class. Then it's going to be very boring because nobody is going to say anything. You will just be sitting over there. Even when you want to solve our own problems, you will just be sitting. You don't want to. You won't participate in doing anything. Okay, so take a very brief overview of the course. Even though it's the name is industrial biochemistry, you could see that there is some interchange of microbiology and biochemistry. So, I mean, generally, these are the things that we'll be looking at. Applications of enzymes. We have looked at enzymes. I mean, last semester we did a lot on enzymes, and then so this time around, what we'll do is to look at how the enzymes can be applied in industry. Then we'll also take a look at different forms of bioreactors, in which you have these fermentation processes occurring. We'll see different forms of these bioreactors, and even look at the factors, or let's say parameters that are controlled in those bioreactors in order to have the optimum yield of the fermentation processes, or let's say the microbiological processes or biochemical processes which are required in the production of a particular product. It could be pharmaceuticals. A lot of times, like say maybe some production of some drugs, or even some biomolecules like some proteins, or even some other enzymes from like production of enzymes from microorganisms. So a microorganism can actually be grown. I mean, within some optimal condition in a bioreactor, and then as it grows, it also produces maybe some particular substance that is needed uh, as a substance to be used for some pharmaceutical products. So we'll look at some of those things. The food and beverage industry, particularly the brewery, the dairy industry. I mean, milk, production of milk, cheese, yogurt, food production, like in the bakery, bread, for instance. Uh, Something also to do, biofuels, mostly bioethanol. How this is done, and then some waste management applications of microorganisms in waste management, what we even refer to as bioremediation. So in a way, maybe some pollution that has occurred either in water and in soil, and you just make use of microorganisms to break down a pollutant, so to make it less toxic or even non-toxic, because once it's broken down into some other substance which is non-toxic, then you would have sort of bioremediated either the land or that water that has been polluted. Now we we did so, like I was saying, we did look a lot at enzymes as biocatalysts that speed up chemical reactions. And so you want to go back and revise those things. The simplest of the things we did about naming of the enzyme. So when you see the enzyme, you can know what it does. Categorization of these enzymes into different classes. Uh, those that catalyze oxidation reduction reactions, those that catalyze transfer of functional groups from one molecule to the other, those that catalyze hydrolytic reactions, the breakdown of big molecules into smaller molecules with participation of water, those that uh, catalyze reactions of either addition of groups uh, to remove double bonds or the other way, the removal of groups that will result into formation of double bonds in a molecule. Those that catalyze isomerization, changing of a molecule into its isomer, and then those that also catalyze reactions of joining molecules together, synthetic reactions, biosynthetic reactions, the ligases, and also things like the specificity of enzymes. Uh, we did refer to absolute specific, group specific, bond specific, looking at things like coenzymes, cofactors, all of those things. You need to revise them so that you can have an understanding into the application of these enzymes. I mean, practically, if before you can understand uh, how these enzymes are acting in industry, you need to have that, theoretical um, foundation. Without a theoretical foundation, there's no way you will understand it. You'll just be seeing it as something is happening, or you just know that this is added to this, and when you are preparing this or that, that's all. And that, if it is that way, then you are not a specialist. All right. And so I mean, some examples of these things are like, enzyme like amylases and proteases. So we've seen amylases to be, enzymes that break down carbohydrates, and this is applied in, say, in bread making, for instance. Where you have say yeast, for instance, that is introduced into uh, the baking of bread, and as the yeast breaks down carbohydrates, it produces carbon dioxide as a byproduct, which will result into the rising of the bread, something like that. Proteases used in the production of cheese break down some proteins, and then also to form some thick curds in that process. How enzymes such as proteases, lipases. What are lipases? Enzymes that break down lipids, I mean fats and oils. These are used in detergents. So you see, detergents that are supposed to be for stain removal. And some of these stains are oils. And so some of these detergents are embedded with some lipases, for instance. And those lipases help in the breakdown of the lipids, like the fatty acids, uh, so that the stain can easily be removed from a textile that is being washed. For instance, this of course has an advantage of doing this at a low temperature, which is more eco-friendly than to be using high temperatures in washing, like using very hot water to be washing. Instead of that, like washing machines, earlier washing machines, heated up water to some high temperature to remove stains from clothes. And then engineering of these detergents, over the years had incorporated some of these enzymes so that same process of removal of stains can be done at a lower temperature. Because the water which is being used to wash in the end is going to be released into the atmosphere. So if you have these enzymes that are being used at lower temperatures, that is much better. I mean, to conserve energy, the machine will not have to heat the water before it uses it to wash and things like that. Then you have uh, enzymes also applied in pharmaceuticals. I think we even looked at some of these examples when we were looking at enzyme kinetics and how it is applied in drug manufacturing or drug discovery, where based on the activity of a particular enzyme, a drug is produced targeting that particular enzyme, its activity, inhibiting it. Similarly, things like diagnosis of some conditions. Some, I think, we looked at an example of um, a device for testing um, fasting blood sugar, for instance, which you know it was established on the activity of two enzymes, hexokinase and glucokinase. So I mean, we did that just last semester. So if you go back and look at some of those slides, you will see those things there. Biofuels in the production of say bioethanol, for instance, you have enzymes like cellulases that will break down cellulose, the indigestible part of, the fiber of the plant material, the biomass, which is being used to produce the ethanol. Another, enzymes that could break down the um, carbohydrates, say, uh, sort of this oxidation reaction that will result into the production of ethanol in terms of anaerobic um, metabolism. You do remember that. So that is what you refer to as fermentation. Similarly, in textiles, I mean, if you use cotton, for instance, cotton, to you in what is referred, like processing and what is referred to as biopolishing of this material. Enzymes like cellulase are also used, uh, which will break down these cellulases, and which will break down cellulose so that you have a more polished fabric. You have something, what you refer to as, okay, in leather making, for instance, you have dehairing and baiting. Baiting is an interesting one. That is in the process of leather processing. What happens is that in the past, how these things were being done was like they like in the shoe manufacturing industry, in the leather industry, they used very uh, you know things like excrement of dogs and pigeons uh, to soak the leather. They soak the leather because these excrements contain enzymes that can break down proteins, and that will result into softening of the leather. you know but that is so a long time ago. Now that process is very well refined because uh, industrially those enzymes can be produced, and so that the leather can be treated with the enzymes to soften them, and then you can have that polishing of the leather. So that is an interesting thing. You have bioprocessing uh, in different forms of bioreactors, which we will look at this. We will look at the different kinds of bioreactors, what we refer to as, the batch system, uh, where you load your raw materials and the system is closed and you just have to wait up to the end. Like if it is going to take an hour or 2, 3, 4, 5 hours for the whole process to come to an end, you have to wait till that time, to get a product, to harvest the product. And we'll take a look at more modern processes as the fed batch system and the continuous system. Fed batch, in which uh, along the line you can have feeding of the system. You can introduce feedstock at a point, apart from, say, the beginning. You can introduce feedstock along the line after a certain stage, and then wait till the harvesting time for you to get a product. Then you have systems the very modern systems that work now are the continuous systems. These ones, have uh, bioprocessing, systems that will have the feeding in of feedstock at a point. Then there is processing ongoing, and at a certain time, harvesting starts. But as harvesting starts and can be ongoing, there can still be feeding in of feedstock for the processing to be carried out and harvesting. So harvesting can be done continuously because the processing of the feedstock, which is also being fed in continuously, is ongoing. This is actually it's a modern system which is being used now industrially because this is more economical. Uh, it is more sustainable, especially with the rising populations. If you want to be producing enough, products, of course, the continuous system is what is convenient to be used. Then there can also be some very specialized systems. Also, depending on a particular product which is being produced, various industries will have some specific systems which they use. These are engineered by multidisciplinary teams. You can have a biochemist, a microbiologist, a physicist, all these people, and industrialists, engineers. coming together to put a whole system, to build a whole, system together. That's like how you put up things like a complex refinery. It's a work which is done by a multidisciplinary team. Just one specialist or one group of specialists cannot do these things. So we'll see. This is just a diagram or an example of a biodigester to see how these things work, where you can clearly see that there is an entry point of the feedstock, sort of you have feeding in, so it's the feedstock is pumped into that digester, and in the digester also you have an entry for air, because of course microorganisms would need some if it's some aerobic system, there must be some oxygen coming in for the microorganisms to grow. And then you have this agitation system. This rotary will have to be turning to have a uniform mixing of the reaction mixture continuously. Obviously, you need to have some monitors. These things are computerized. Now it's not a thing that is manual. So you just key in, say what temperature, what pH, what pressure, because this you have some sensors that are inside, the reactor to monitor these things. If there is anything like maybe rise of temperature too high or anything, quickly the sensor is going to give a signal, and if you have to stop the system or quickly if you have to attend to it in a way, all those things you can easily do them just by using buttons on the device. So it's not anything which is complex that you cannot do once you have an orientation in your workplace where you are going to work, and that's it. You can easily do it. Then, of course, there is the exit. So where you see effluents, that is where you have the harvesting of the product coming from. After you harvest, sometimes you need to also you have further processing, because the harvesting may still have some impurities in it, and so there will be some other processes of purification and things like that to get the pure product which is being produced in the reactor. All right, but this is just an overview, so we'll take a look at it much more. I think I had some videos. I will look through my things. If some of these videos are there over the period, I will share some of these. I hope I can get maybe some links of these videos and send to you so you can watch them and see these things in reality, how they are done in industries. Then obviously, you did remember that we looked at factors that affect enzyme activity. We talked of say temperature, pH, even concentration of substrates, stuff like that. This time around in the industry, yes, same parameters like temperature, pH need to be controlled in order to have the maximum yield of a product. Because especially if you are having a microorganisms that are producing some enzymes or that are having some enzymatic activity on a product, breaking it down, obviously you require an optimum temperature at which these microorganisms will act. Similarly, you require an optimum pH for the process to be carried out. Oxygen, as we just talked about it, the process, requires a certain optimum concentration of oxygen, so that if it's not controlled, it may result into maybe clogging of the system. For instance, maybe microorganisms could get overgrown, for instance, or maybe uh, not enough oxygen in the system. Instead of an aerobic process ongoing, you rather be having some anaerobic process, So, or if it is an anaerobic process, which should occur, say if you need to have uh, fermentation of carbohydrates to produce ethanol. Obviously, you need to have an anaerobic condition. But if you're having too much of oxygen in the system, then you have your carbohydrates. What will happen? It will just, break down and be producing carbon dioxide gas, water, and you will not have your ethanol, which will be produced. Agitation, that is the mixing of the reaction mixture. So there should be maybe what we call uh, the speed of the the rotary there, where you have maybe a certain revolution per minute. That too have to be controlled to have homogeneous mixture at all times, so that you have um, the reaction occurring, at all parts of the system evenly. Right. Again, you also want to remember, things like inoculation you did in microbiology, and you want to remember. I think we'll look at it briefly after this. That growth curve that you've always been talking about in microbiology, because you are going to refer to it in engineering of systems. If you know the growth pattern of a particular microorganism, then say, in which amount of time the microorganism would have reached, uh, its stage where it can produce the product that you are looking for. All right, so I think I talked much about this aerobic and anaerobic respiration already. And yes, the fermentation process, you first, microorganisms will first have to get inoculated. Sort of, it's like a nursery. You grow the microorganism in a smaller, uh, container, maybe in a solution, before you transfer it into a bigger, uh, digester, where now growth can, because now you have a bigger space, the microorganisms which you have inoculated now can grow, can reproduce. And then once they multiply, then they will begin to now produce the product what you are looking for in larger amounts. And then you can now do the lasting harvesting. Well, not the lasting per se, because even after harvesting, like I mentioned earlier, you can have the product having some impurities. So there can still be some processing after that, say purification processes, to get a pure product out of it. Yeah, so this is what I was talking about, the microbial growth curve that you want to revise. You've done it in microbiology over and over again, where you have over a certain period of time for whichever microorganism that you are studying, you have that microorganism going through different growth stages, what we refer to as the lagging stage, where after you do the inoculation of the microorganisms. So there is that very slow growth, and then it reaches a point where you have these microorganisms begin to multiply in numbers. And then there's that stationary phase. So what I was saying is that if you want to make use of any microorganism in industry, you need to understand this growth pattern of that microorganism to know when the microorganism is going to reach a stationary phase. Because it is at this stationary phase that you can have your products being produced in optimum amounts. At the stage of exponential growth, the microorganisms at this point are multiplying in numbers. And once they reach stationary phase, means now they have reached their maximum quantity. That's now the whichever biochemical process which should be taking place will be constant at this point to give you the optimum yield that is needed. And once the stationary phase is over and bacterial death starts, the number of microorganisms begin to reduce at this point. So you should know that at the end of the stationary phase, you should start to do your harvesting so that you don't have dead cells just sort of clogging the system and even making your system even more impure and things like that. So you need to understand this for each and every microorganisms that you decide to use in a industrial system. Then a very simple principle, which perhaps you haven't paid attention to, is that when you take any biomolecule, the biomolecules which we have studied so far, like carbohydrates, proteins, lipids, nucleic acids, like the DNA, RNA. If you pay close attention, you realize that all these biomolecules consist of some major elements. These are carbon, hydrogen, oxygen. You see, this is constant because if you see all these, if you take a carbohydrate, the monosaccharides like glucose, fructose, all of them, don't you see those structures contain carbon, hydrogen, oxygen, mainly. If you take lipids, the fatty acids, the same thing, carbon, hydrogen, oxygen. If you take proteins, so in proteins, you find nitrogen because you're having an amine group there. The same way, if you take the nucleic acids, you see there too, apart from the carbon, hydrogen, oxygen, there will be some nitrogen because you have a nitrogenous base, which is on a pentose with a phosphate group. I said a phosphate group. So that is phosphorus also coming in there. So in fact, if you take biological systems, the major components you will find will be carbon, hydrogen, oxygen, nitrogen. You'll find sulfur also because you remember that you have some of the amino acids containing sulfur. At least you can remember two: methionine, cysteine. And you see phosphorus too, you find it in the nucleotides that form the nucleic acids. So these think 5 or 6 elements: carbon, hydrogen, oxygen, nitrogen, sulfur, phosphorus. These are major components that you will find in uh, biomolecules. And also in industry, these particular components are nutrients which you will need to feed into your reactor for the microorganism to grow, because the microorganism needs this to grow in order to produce the product that you want. So there are some feedstocks that are depending on a particular product which you are producing and a particular microorganism which you are producing, the system which is being used. These industries have some specific feedstocks that they feed into these systems, which are, rich in carbon, hydrogen, oxygen, and perhaps things like nitrogen, phosphorus, sulfur, depending on a particular product that it is they are producing. These are the other nutrients which they also introduce into the system at different uh, ratios, different combinations. Right. Okay. So, I mean, this is to look at some specific example of these things we talk about with the enzymes, for instance. So you have, say, maybe in the dairy industry, let's say in the production of, say, yogurt, where there's supposed to be some fermentation of milk. Specifically, what is being fermented there is lactose, which is milk sugar. So lactose is being fermented by lactobacillus, and so that lactose breaks down to smaller molecules from lactic acid, acetaldehyde, diacetyl. Particularly lactic acid, acetaldehyde will give the sour taste which you'll observe in yogurt and some other milk products. Or you have but this process of the lactose broken down into lactic acid, this is occurring within the organism, within the lactobacillus. But the lactobacillus also can release some extracellular proteases which will act on proteins, say the milk protein, casein. So the lactose is milk sugar, but casein is protein, is milk protein, so that the activity of such extracellular proteases will result into, the formation of some thick curds due to the breakdown of casein into, some shorter peptides and some amino acids. You can also see another microorganism, a thermophilic streptococcus, which also carries out similar reaction of, the breakdown of lactose into lactic acid, acetaldehyde. And you see in these processes we have seen this coenzyme, folic acid, which is required by these microorganisms in carrying out these enzymatic reactions. Of course, you also see that there can be some waste product which is produced there, which is ornithine. And you do remember ornithine was one of those intermediates which you find in the urea cycle. So if you go back to our studies on enzymology, revise, you will see this, you will see these things. All right. So that's just some example there. Another one you talk about is in the production of, say, bioplastics. And you can see something like lipases, which act in the breakdown of lipids like the triacylglycerols into, shorter chain hydrocarbons and their esters. So you see that, or in the production of different forms of bioplastics, those with some aromatic chains, I mean some polyolefins, even the ones with some halogenated groups. All of these have some applications of enzymes in their production, even in the production of things like bioethanol, biodiesel that we talked about much earlier. Production of bioethanol, largely is from sugarcane. I think the country in the world that produces more of this bioethanol is Brazil. I think they are the biggest in terms of bioethanol production. They have these large sugarcane farms, which is economical. There's, I think I just remember something. Some years back, there were some Americans, I think, yeah, there was some American company that came to Ghana and was wanting to do some biodiesel production from a plant, a local plant we have in Ghana, Jatropha. I don't know if you know, there's this plant, Jatropha, which people just grow it like a fence plant. Like it's a shrub, it's not a big plant. And it produces some seeds. You can later Google Jatropha and you will see. I'm sure you know it. Maybe even if you don't know the name, if you didn't know the name, if you type and you see the picture, you see what I'm talking about. And I mean, in the villages, what people were doing, and in playing with these things, they take the seeds, dry these seeds, and put it on broomsticks, and they light it, in the night. And I think there was a Ghanaian in America sometime who took interest in studying that plant, the seed, to see what was in it that was causing it to burn, and realized that there were actually some hydrocarbons in there, like sort of some biofuel, and so thought that they could do some extraction of biodiesel from the seed. And so this company came to Ghana and came to uh, establish some large plantation somewhere, I think around Adan, thereabouts. But I think along the line it was abandoned, and they just left. And that is understandable. I think sometimes when you have some scientific ideas and they look beautiful, but they may not be practicable. So, for instance, if you compare that plant, that you have to harvest seeds, dry them, or maybe the seeds are already ripe and dried before you are harvesting them. Now, how are you going to harvest the seeds? You need, if it's a plantation, a large plantation, you need a large number of people to get into the plantation to be harvesting those seeds. I mean, compare that to, say, sugarcane, that the Brazilians will grow a plantation of sugarcane. When the sugarcane is mature, they just send in a combine harvester and to weed off everything, just harvest everything with the the stems, the leaves, even some grass which is in it. It doesn't matter. All that, harvest it like some biomass, and then they just go send it into processing, mash it, and all of that, and uh, provide all the conditions for fermentation of the sugar that is in the sugar cane, and they produce different products out of it: sugar, the bioethanol, the fiber itself too. They can process it, produce other things out of it. So that is much easier, more economical. You can easily do that. But if you have to, the plant, the jatropha, you can't weed off everything and replant it. It's going to take a long time for it to grow before it starts to seed again. And so the plant has to be there, and you'll be harvesting only the seeds. So it's not, it wasn't economical. So I mean, yeah, just left it and went away. So that is to see. Sometimes if you have some scientific ideas, beautiful on paper, but they may not be practical. All right. So we did talk. We also said applications of this uh, microorganisms also in wastewater treatment. So where you can have solid waste or wastewater that, as you see, comes into waste management plants. Goes to a point where you have separation of the solids from that wastewater, and then the wastewater goes back, for treatment into fresh water and can be pumped back into the system. In fact, in some countries, all waters that are used, almost all, from say residences, for domestic use, are recycled and goes back into the system. Because once you have a separation of solids from it and some purification systems and things, disinfection and all of that, the water becomes wholesome again to be used again. So there's no waste in the system. And it is the use of these bioreactors with some uh, uh, applications of microbiology in these things, in those reactors, that makes it possible for such cycling of water. And then I think this should be the last thing we talk about. Um, even though we talk of this, biological process as being sustainable, eco-friendly, and all of that, there could be some issues with scaling up of them. I mean, an example I was giving you was this Jatropha thing, which, I mean, if you want to have such a thing, you need a large number of people. How much are you going to pay each and every person that's going to be on that plantation? And how much of the product are you going to produce from those seeds that you are harvesting that will actually, pay for all those expenses in order for whichever business, that business of production of biodiesel to be sustained. And so that's clearly an issue with scaling up of such a technology. The use of uh, biofuels, in countries, you have some regulations of the percentage of these biofuels that can be used, to mix, fossil fuels. So you have a different, combination, different percentage of biofuels that are allowed in these fossil fuels, It's also an economic question. I mean, to sustain the economy of countries and things like that. Sustainability, yes, I mean, this uh, has to do with the maybe some toxic wastes which can be produced in some of these processes. The practicality of this of the technologies that are employed. So anytime someone or any group of scientists are designing any new technology, of course, these are things that have to be considered: sustainability and environmental um, impact of the technology that is being introduced. Of course, some ethical considerations are considered when it comes to, say, genetic modification of some of these microorganisms in order to have a increased yield. Uh, will some of these genetic modifications not result in some strains of bacteria that later will become harmful? I mean, all of these are questions that need to be considered in the development of um, technologies to be used in industries. All right. So at this point, I thank you for your audience, and if you have any questions, like as I said earlier, you can write in the chat. Um, Alternatively, you can raise your hand. I have enabled all your microphones, so you can unmute yourself and speak, and I will respond to you. All right. So the floor is open.